There was a paper I found on a Tuesday night, sitting at my desk at around 11 o'clock, when I should have been doing something else entirely. And it stopped me so completely that I closed everything else I had open and just read it twice through without moving. It wasn't a dramatic paper. It wasn't written in a way that was trying to get anyone's attention. It was a study published in the journal Nature, Ecology and Evolution in 2018, and it was describing the stomach contents of amphipods, small shrimp-like crustaceans, that had been collected from the Mariana Trench at depths between 7,000 and nearly 11,000 meters below the surface of the Pacific Ocean. What the researchers found in those stomachs, which the paper's authors, Alan Jamieson and his team at Newcastle University, described in careful, restrained scientific language, was persistent organic pollutants, polychlorinated biphenyls, PCBs, the same industrial chemicals that were manufactured, widely used, and then banned in most countries by the 1980s because of how toxic they are. Banned because they do not break down. Banned because they accumulate. In the stomach tissue of animals living at the bottom of the deepest place on Earth, roughly 50 times more concentrated than in crabs living in heavily polluted river systems in China, there were the chemical traces of a world those animals have never seen and have no framework for. I sat with that for a while before I could move on. Not because it was the strangest thing in the paper, not because it was the most scientifically significant finding, though it was significant. I sat with it because of what it implied about the relationship between the surface world, the world you and I inhabit, the world of factories and ships and rivers running to sea, and the deepest, most remote, most inaccessible place on our planet. The implication was that there is no separation, that what we do up here arrives down there, eventually, carried by currents and marine snow and the slow settling drift of particles through 11,000 meters of water. The trench does not know it is remote. The trench does not have walls. That was the question I could not put down. Not just what lives down there, though that question is genuinely one of the most interesting in all of biology, but something larger and more unsettling. The question of what it means that we have spent our entire history on the surface of this planet, looking up at stars and sideways at continents, while directly beneath us, under a layer of water deeper than any mountain is tall, there has been a world we have visited fewer times than we have visited the surface of the moon. A world that is, by volume, the largest habitable space on Earth. A world that has been operating according to its own rules, under pressures and in conditions that should not, by any biology we understood before 1977, support life at all. The Hadal Zone. The word comes from Hades, the Greek underworld. It was coined in the 1950s by the Danish marine biologist Anton Bruun, who led an expedition called the Galathea Expedition and came back with animals collected from depths greater than 6,000 meters. Animals that should not have been there by the scientific understanding of the time. Animals that suggested the bottom of the ocean was not the silent, empty desert that most researchers of that era assumed it to be. He needed a name for the zone. He reached for the underworld. You can understand why. I want to spend tonight in that zone. Not to make it frightening. It is not, or at least the fear is not the interesting part, but to describe it as precisely as I can, to tell you about the people who have actually descended into it, and what they found and what it cost them to get there, to spend some time with the organisms that have made it their home, and the extraordinary biochemistry that makes that possible, and to be honest with you about how much we still do not know, which is most of it. The Hadal Zone is by a very significant margin the least understood major habitat on our planet. According to figures from NOAA's Ocean Exploration Program, which tracks ocean floor mapping coverage, we have better maps of the surface of Mars than we have of the deepest trenches on Earth. That fact alone, I think, is worth a night. You are lying somewhere specific right now. The room around you has its particular darkness, its particular temperature, its particular weight of silence. Your body is heavier than it was a few hours ago, the accumulation of the day settling into whatever surface is beneath you. Something is probably still running in the background of your mind tonight, the way something always is, some unfinished loop from the hours before you came to rest. That is fine. You do not need to resolve anything tonight. You do not need to hold on to what I am about to tell you. This is not a test. Nothing here needs to survive until morning. Just let it come through, at whatever depth it reaches, and if you drift off somewhere between the surface and the bottom, that is exactly where I hoped this would take you. Let me start with the shape of the place. The ocean covers approximately 361 million square kilometers of the Earth's surface, roughly 71% of the planet you are lying on right now. It has an average depth of about 3,600 meters, which is already a number that requires some pause. If you could lay the Eiffel Tower on its side, nearly 12,000 times end to end, you would cover the average depth of the ocean. But the average is misleading in the way that averages of extreme distributions always are, because the ocean is not uniformly deep. It has shelves and slopes and basins and ridges, and at its most extreme, trenches, narrow, elongated depressions in the seafloor formed at the boundaries of tectonic plates, where one plate is being forced beneath another in a process called subduction, and the crust buckles downward under the weight of that geological collision. Marine scientists divide the ocean by depth into zones, and it is worth moving through them slowly, because each one is a genuinely distinct world, not just a point on a scale. The sunlit zone, the photic zone, extends from the surface down to roughly 200 meters. This is the zone most of us mean when we think of the ocean, the zone where light penetrates, where photosynthesis is possible, where the familiar animals of coral reefs and open water spend their lives. Below 200 meters, the light begins to change. Between 200 and 1,000 meters is the mesopelagic zone, the twilight zone, where sunlight fades from blue to indigo to the faintest violet, and then to nothing. At around 1,000 meters, even the most sensitive instruments designed to detect visible light find nothing. The sun does not reach there, not even a suggestion of it. Below 1,000 meters is the bathypelagic zone, the midnight zone, running down to 4,000 meters. Below that, from 4,000 to 6,000 meters, is the abyssal zone, which covers the vast majority of the deep ocean floor. The abyssal plains, wide and flat and largely featureless, spread across roughly 60% of the planet's surface. And below 6,000 meters, in the trenches, begins the Hadal Zone, the bottom 5% of the ocean, by depth. The place named for the dead. There are approximately 37 Hadal trenches on Earth, according to surveys compiled and published by Jamieson and colleagues across several papers on Hadal biogeography. Some are familiar names: the Mariana Trench in the Western Pacific, the Tonga Trench and Kermadec Trench in the Southwest Pacific, the Puerto Rico Trench in the Atlantic, the Java Trench in the Indian Ocean. Others are less well known: the South Sandwich Trench in the South Atlantic, the Izu-Ogasawara Trench, extending south of Japan, the Malloy Deep in the Arctic Ocean, the Peru-Chile Trench, running along the western edge of South America. Together, these trenches represent a combined environment unlike anything else on Earth. And despite decades of effort, most of them have never been directly sampled at their deepest points. Several have never been visited at all. The deepest of them, the deepest point on the surface of the Earth, is Challenger Deep in the southern end of the Mariana Trench. Its depth has been measured multiple times with improving precision. The most widely cited recent figure, produced by a multi-beam sonar survey conducted as part of Victor Vescovo's 5 Deeps expedition in 2019, and published in the journal Geochemistry, Geophysics, Geosystems, places Challenger Deep at approximately 10,928 meters below sea level, though the uncertainty range of the measurement itself is on the order of several meters in either direction, which is a reminder that even the most basic fact about this place, its depth, is something we are still refining. 10,928 meters. If you placed Mount Everest at the bottom of Challenger Deep, the peak of Everest would be more than 2,000 meters below the surface of the Pacific. You could stack a further 2 Empire State Buildings on top of Everest before you broke the surface. The mountain would be invisible from above. The ocean would swallow it completely and have room to spare. The pressure at that depth is approximately 1,086 atmospheres, a figure cited in multiple NOAA ocean exploration publications and consistent with the calculations of Vescovo's expedition scientists. 1 atmosphere is the pressure of the air pressing down on you, where you are lying right now. A pressure so normal that you cannot feel it, because your body has spent its entire existence calibrated to it. At Challenger Deep, the equivalent pressure is roughly 15,900 pounds per square inch. If you prefer a more immediate analogy, the pressure at the bottom of Challenger Deep is roughly equivalent to having 50 standard-sized passenger aircraft stacked directly on top of a single postage stamp. Every square centimeter of any object at that depth is bearing a force that would, without significant engineering countermeasures, collapse it. Not damage it. Collapse it. The temperature at Hadal depths is typically between 1 and 4 degrees Celsius, just above freezing, cold enough that biochemical reactions slow significantly compared to the surface. The water is completely dark. It has never been touched by sunlight in the entire history of the Earth. What light exists in the Hadal Zone, and there is some, is biological in origin. Animals that make their own. Before we go deeper into what lives there, I want you to notice the weight of your own body for a moment. The surface beneath you. The air in the room at whatever temperature it is tonight. The Hadal Zone I am describing is not somewhere you can go in any practical sense. At the time of writing, the total number of humans who have descended to Challenger Deep is fewer than 30. In a vehicle specifically engineered over years to withstand the pressure, at enormous expense, for brief visits measured in hours. More people have walked on the surface of the moon than have visited the deepest point of the ocean. NOAA's ocean exploration program repeats this fact often, not as a complaint, but as a way of orienting the scale of how little we know. The moon is approximately 384,000 kilometers away. Challenger Deep is under the Pacific Ocean, and yet it might as well be on another planet. You do not need to remember any of this by morning. Nothing gets tested. Nothing needs to stick. Just let it move through you at whatever depth it reaches. The story of how humans first got there begins in a spherical steel vessel hanging from a cable in the Bahamas. In 1930, William Beebe was a naturalist, a curator at the New York Zoological Society, and he was not satisfied with what dredges pulled up from the deep sea. Dredges could collect animals from the bottom, but they could not show you the world those animals lived in. They could not show you what was swimming at 500 meters, or what the light looked like at that depth, or how the animals behaved in the context of their actual environment. Beebe wanted to go there himself. He partnered with an engineer named Otis Barton, who had designed a vessel he called the bathysphere, from the Greek bathos, meaning deep. It was a hollow steel sphere 57 inches in diameter, with walls 1.5 inches thick, with fused quartz windows 3 inches thick. connected to the surface by a single steel cable and a rubber hose, carrying the telephone and oxygen lines. It had no propulsion of any kind. It went where the cable lowered it and came back when the cable pulled it up. If the cable had broken, Beebe and Barton would have sunk to the bottom. On August 15, 1934, Beebe and Barton descended to a depth of 923 meters, just over half a mile, in the bathysphere, off the coast of Bermuda. It was the deepest any human had ever gone into the ocean. Beebe described what he saw through the quartz windows in a book called Half Mile Down, published that same year, and it is worth pausing on some of those descriptions because they represent the first human account of what the deep ocean actually looks like from the inside. He described the light changing as they descended, the sunlit blue of the surface fading through indigo and violet to what he called a color he had no word for, a blackness that was not the blackness of a room with the lights off, but something more absolute, more total. What he called a black blue that pressed against the windows with a weight he could feel even though he knew it was optical. He described fish passing the windows that he recognized from surface trawls as deep-sea animals, lanternfish, bristlemouths, and others he could not identify at all. Animals trailing lights and filaments and structures he had no taxonomy for, some of which he described to his colleagues upon returning to the surface, and was not entirely believed. Beebe had a reputation for slightly heightened description, and some of the animals he reported were treated with skepticism for decades. But the core of what he recorded was accurate. The deep ocean, at depth, is not empty. It is full of things moving in the dark. The bathysphere was not capable of reaching the Hadal Zone. 923 meters is well within the bathypelagic, the midnight zone, and extraordinary by the standards of 1934, but still 7,000 meters above the bottom of Challenger Deep. Reaching Challenger Deep required an entirely different approach to engineering, not a sphere hung from a cable, but a vessel that could navigate under its own power, that could withstand pressures an order of magnitude greater, and that could be recovered if something went wrong. It required a bathyscape. The bathyscaphe, again from the Greek meaning deep vessel, was designed by Swiss physicist Auguste Piccard, who had previously designed stratospheric balloons and understood. By intuition refined over a career in extreme engineering, the principle of using a buoyancy float to control vertical position in a fluid medium. His design was elegant in an engineering sense. A large float filled with aviation gasoline, which is less dense than seawater and therefore provides positive buoyancy, with a small, heavily reinforced pressure sphere attached beneath it where the occupants would ride. To descend, you released iron-shot ballast, increasing the overall density of the assembly. To ascend, you released more shot. The gasoline kept the assembly buoyant enough to return to the surface when all the ballast was gone. The pressure sphere, developed with Auguste's son Jacques Piccard in collaboration with the United States Navy, had walls of high tensile steel more than 3 inches thick. Its single observation window, a cone of synthetic forsterite, a material chosen for its ability to transfer compressive loads to the surrounding steel rather than bearing them directly, was 5 inches thick at its outer face and 3 inches at its inner face. The window was not round, it was conical, because a cone under compression transfers load into the surrounding structure, and that design choice is the reason the window did not collapse on January 23, 1960. That was the day Jacques Piccard and United States Navy Lieutenant Don Walsh descended to the bottom of Challenger Deep. They left the surface at 8:23 in the morning. The descent took nearly 5 hours, at a rate of approximately 1 meter per second, slow enough to allow the structure to adjust to the pressure gradient, slow enough that the gasoline flowed above them could equalize its own internal pressure through valves. Walsh, in interviews he gave over the following decades, he died in November 2023 at 92 years old, the last surviving member of the first human descent to the deepest point of the ocean. Described the view through the window as the descent deepened as an absolute darkness into which their lights reached only a short distance, perhaps 3 to 5 meters. And beyond that, nothing. No walls, no floor. Just the cone of their own illumination and the darkness pressing in from every direction. At 9,100 meters, the outer plexiglass pane of the entry tunnel, not the main observation window, but a secondary structural element, cracked. The sound, as Walsh described it, was like a gunshot resonating through the entire hull. Picard and Walsh looked at each other. The hull integrity gauges showed no compromise to the main sphere. They continued descending. At approximately 10,912 meters, the depth, as registered by their own instruments, which later analysis suggests was within a few hundred meters of the actual bottom, the Trieste settled into the sediment at the floor of Challenger Deep. The time was 1:26 in the afternoon. They had been in descent for nearly 5 hours. They had no cameras capable of capturing the scene in meaningful detail. They had no robotic arms to collect samples. They had one observation window, a depth gauge, and a telephone line running back to the surface support ship. Walsh later said that what he saw through the window, as the cloud of disturbed sediment slowly settled around them, was a pale flatfish, a sole or something resembling one, lying on the bottom, apparently alive and apparently unbothered by their arrival. That observation became contested. Marine biologists who studied the dive subsequently argued that the animal Picard and Walsh saw was more likely a sea cucumber or possibly a type of large polycake worm, both confirmed Hadal residents, rather than a flatfish, because the subsequent 60 years of Hadal biology have suggested strongly that vertebrates with swim bladders cannot survive below approximately 8,400 meters, for reasons I will describe shortly. But Walsh maintained his account for the rest of his life. He had been there. No one alive could contradict him from personal experience. They spent 20 minutes on the bottom. Then they released iron-shot ballast and began the ascent, which took 3 hours and 15 minutes. They surfaced in the early afternoon, in the Pacific Ocean, having done something that no human had done before and that no human would do again for 52 years. That gap is worth dwelling on. 1960 to 2012. 52 years during which humans launched probes to the surfaces of Mars and Venus and the outer planets, built space stations, landed on the moon 12 times, sequenced the human genome, and developed the internet. 52 years during which the deepest point of our own ocean, under the Pacific, accessible by ship, was visited by no one. Part of the reason was cost. The engineering required to safely reach Challenger Deep is genuinely expensive, and the scientific return per dollar compared unfavorably with unmanned landers, which can collect specimens and measurements at a fraction of the cost of a crewed dive. Part of the reason was risk. The crack in the Trieste's outer window is a reminder of how narrow the engineering tolerances are at that depth, and the history of deep-sea submersible operations includes complete vehicle losses. One of them was Nereus. Nereus was a hybrid remotely operated vehicle designed and built by the Woods Hole Oceanographic Institution, one of the world's leading oceanographic research organizations and the home of Alvin, the workhorse crewed submersible that has made more dives to the deep ocean floor than any other vehicle in history. Nereus was designed specifically for Hadal research. It could operate either as a remotely operated vehicle, guided by a fiber optic tether from the surface, or autonomously without a tether, diving to full Hadal depths and operating for hours. It made its first successful dive to Challenger Deep in 2010, collecting samples and high-definition video footage from the bottom. It subsequently made dives to other Hadal trenches, gathering biological and geological data. On May 10, 2014, during a dive to the Kermadec Trench, northeast of New Zealand, reaching a maximum depth of approximately 10,047 meters. Narration imploded at approximately 9,900 meters. The pressure crushed the vehicle's pressure housing instantaneously. No one was aboard. The surface team recovered only small fragments of yellow foam and circuit boards floating where the vehicle had last been tracked. Weihai's documentation of the loss attributed it to a fatigue failure in a pressure-rated component, a piece of material that had been stressed and flexed over multiple dives until it reached its limit at the worst possible moment. $8 million. Years of engineering work, gone in an instant because of a piece of material slightly smaller than your hand. The field of Hadal research accepts loss in a way that most sciences do not have to. The pressures involved mean that equipment loss is simply part of the operating conditions. The science continues because the alternative is not doing the science. James Cameron arrived at Challenger Deep on March 26, 2012, in a vehicle of his own design called the Deep Sea Challenger, which was in several ways unlike anything that had preceded it. Where the Trieste was primarily a buoyancy system, a large float with a small sphere, the Deep Sea Challenger was essentially a vertical torpedo, a 12-meter-long cylinder, bright green, with a pressure sphere embedded at one end. The sphere was 67 centimeters in diameter, small enough that Cameron could barely move inside it, and the vehicle was designed to descend nose down, which reduced its hydrodynamic profile and allowed significantly faster descent and ascent than the Trieste had managed. Cameron had spent 7 years working with a team of engineers to build it. It carried 11 hours of battery life, a 3-dimensional camera system designed specifically for the dive, robotic sampling arms, and thrusters for horizontal maneuvering at depth. The descent to Challenger Deep took 2 hours and 36 minutes. Cameron reached a depth of 10,908 meters, as measured by his instruments. He spent approximately 3 hours on the bottom, moving horizontally across the sediment under thruster power, collecting samples of sediment and water, filming with his camera systems. He described the bottom, in subsequent interviews, as a very lunar, barren place. He said he had expected more visible life, more organisms moving across the sediment, more immediate biological activity. But what he primarily encountered was sediment. Pale, soft, fine sediment, undisturbed until his thrusters passed over it and then briefly clouded, and then still again. He found no large animals moving across the bottom during his 3 hours there, though his sediment samples, when analyzed at the surface, were full of microbial life, confirming what lander-based science had been accumulating for years. On ascent, the starboard thruster failed. Then, the port thruster. The vehicle lost its trim. Cameron managed the ascent manually, adjusting ballast to compensate, and reached the surface after 70 minutes. The Deep Sea Challenger was subsequently damaged in transit by rough weather and was never dived again. Cameron donated it to the Woods Hole Oceanographic Institution, where it remains. The third era of human descent to Challenger Deep, the era that has produced the most scientific data and the greatest number of crewed dives, began with a retired American private equity investor named Victor Vescovo, who had a specific and somewhat unusual goal: to reach the deepest point in each of the 5 oceans in a single expedition: the Atlantic, the Southern Ocean, the Indian Ocean, the Pacific, the Arctic. 1 dive per ocean to the deepest confirmed point. He called it the 5 Deeps Expedition. To accomplish this, Vescovo commissioned a new submersible from Triton Submarines in Florida. The resulting vehicle, named DSV Limiting Factor, was in several ways the most capable crewed Hadal vehicle ever built. Designed from the start not for a single record dive, but for operational reliability across multiple deep dives in varied conditions. Its pressure sphere was machined from titanium 140 millimeters thick to tolerances of fractions of a millimeter. It was rated to full ocean depth, meaning it could theoretically be operated anywhere in the Hadal Zone. Vescovo worked with the expedition's lead scientist, Alan Jamieson of Newcastle University, and with geologist Heather Stewart of the British Geological Survey to design the scientific program for each dive. The 5 Deeps expedition ran from late 2018 through 2019. Vescovo reached the Puerto Rico Trench in the Atlantic, approximately 8,376 meters, the deepest point in the Atlantic. He reached the South Sandwich Trench in the Southern Ocean, approximately 7,434 meters, at a location subsequently named Factorian Deep. He reached the Java Trench, or Sunda Trench, in the Indian Ocean, approximately 7,187 meters. He reached the Malloy Deep in the Arctic Ocean, approximately 5,550 meters. And he reached Challenger Deep in the Pacific, which he dived multiple times, accumulating more total time at full ocean depth than any human in history. The depth measurement, produced by the 5 Deeps Expedition Survey Team using a multi-beam sonar system of exceptional resolution, placed Challenger Deep at 10,928 meters, with an uncertainty of approximately 10 meters. That is, as of the current scientific literature, the best measurement we have. What Vescovo found at the bottom of each trench, in terms of immediately visible biology, was in many ways similar to what Cameron had described: sediment and the occasional organism. At Challenger Deep, he found snailfish, several of them, pale, translucent leperid fish, moving across the sediment at depths that should, if you apply the physics of fish biology as understood before the 90s, be impossible for a vertebrate to survive. He found amphipods, the small crustaceans that, in certain Hadal trenches, occur in densities that surprise every researcher who first encounters the footage. He found, at the very bottom of the deepest place on Earth, a plastic bag and what appeared to be candy wrappers. That last item requires a moment. This was not a surprise to Alan Jamieson, who had by that point been documenting anthropogenic contamination in Hadal trenches for several years. The same contamination he described in that PCB paper published in Nature, Ecology and Evolution. But it was documented on camera at the bottom of Challenger Deep, and those images circulated widely, because there is something about seeing human-made debris at 10,900 meters that communicates the reality of the situation more directly than any graph of pollution concentrations. The trench is not remote in any meaningful material or chemical sense. Everything that enters the ocean eventually arrives there. If this kind of thing helps you rest, if a quiet voice following these questions through the dark is something that settles your mind, then sharing it, or subscribing so that more nights like this one find their way to you, means someone else who needs this can find it too. That is the only reason I mention it, and that is all I will say about that. Now I want to tell you about the animals, because the history of humans descending is one story, and it is a story about engineering and courage and money and the particular kind of loss that happens when a machine you built implodes at 9,900 meters because a component reached the end of its tolerance. But the older and stranger story, The story that has been running for tens of millions of years in the dark and the cold and the pressure, without any reference to us at all, is the story of the organisms that live there. That story requires some biochemistry, and I am going to try to make the biochemistry as concrete as I can, because it is genuinely one of the more remarkable things I encountered in all of this research. The fundamental problem of living at Hadal depths is pressure, not darkness. The Hadal Zone has its own darkness, and many of its organisms have adapted to it as thoroughly as any deep-sea animal has. Not cold. The temperatures are cold, but well within the range that biochemical systems can be calibrated for. The pressure is the problem, and it acts in ways that are more fundamental than the simple mechanical compression we tend to imagine. A protein, any protein, including the enzymes that catalyze the chemical reactions that keep cells alive, is a folded structure. Its function depends entirely on its shape, and its shape depends on relatively weak non-covalent bonds that hold the folded molecule in its working configuration. Under extreme hydrostatic pressure, those bonds are disrupted. The protein unfolds. An unfolded enzyme is a non-functional enzyme. Non-functional enzymes mean non-functional cells. Non-functional cells mean dead organisms. This is why, if you placed a surface bacterium at the bottom of the Mariana Trench, it would simply stop working. The pressure would denature its biochemical machinery before it could do anything else. But the bacteria, archaea, and larger animals that live at Hadal depths have not stopped working. They are operating normally, by their own standards, in their own context, at pressures that would destroy a surface organism at the molecular level. How? The answer, at least for the larger animals, the fish, the crustaceans, the echinoderms, involves a small organic molecule called trimethylamine oxide, or TMAO. This molecule has been studied extensively by Doug Bartlett at the Scripps Institution of Oceanography in San Diego, and by Paul Yancey at Whitman College, whose work on the biochemistry of deep-sea adaptation has shaped the field for decades. A paper by Yancey and colleagues, published in the journal Science in 2014, established a relationship that is, in its own quiet way, one of the more elegant findings in deep-sea biology. TMAO is not a structural molecule. It does not become part of the organism's physical architecture. It is what chemists call a piezolite, a pressure-counteracting osmolyte, a small, water-soluble compound that accumulates in the cells of deep-sea organisms and that appears, through mechanisms still being investigated in detail, to stabilize protein structure against the denaturing effects of pressure. It acts, in a loose analogy, like a molecular brace that holds folded proteins in their correct configuration, even when the physical conditions around them are working to push the shape apart. What Yancey's team found was that in deep-sea fish, the concentration of TMAO in muscle tissue increases linearly with depth. The deeper the fish lives, the more TMAO it carries. The relationship is consistent enough across species that you can use a fish's TMAO concentration as a proxy for the depth at which it lives, a biological depth gauge written in chemistry, readable in any tissue sample, regardless of where the sample was collected. For every increase of 1,000 meters in depth, the TMAO concentration increases by a predictable and measurable increment. This matters enormously for understanding the depth limit of fish life, which is one of the genuinely unresolved and actively debated questions in current hadal biology. The deepest fish ever reliably recorded was a snailfish. Pseudoliparus weyeri, the Mariana snailfish, filmed at 8,336 meters in the Mariana Trench in 2017 by a lander operated by a team from the Chinese Academy of Sciences. Prior to that record, The depth record for any vertebrate had been held by a different snailfish species observed at 8,145 meters in the same trench. In 2023, during an expedition to the Izu-Ogasawara Trench south of Japan, a team from the University of Western Australia led by Alan Jamieson filmed snailfish at approximately 8,336 meters, matching the Mariana record. Why snailfish? The snailfish family, Leporidae, appears to be the group that has most successfully colonized the Hadal Zone at the vertebrate level. They are not large animals. The Hadal snailfish are typically between 15 and 25 centimeters long, pale or translucent, with no swim bladder, which is significant because a gas-filled swim bladder, which most bony fish use for buoyancy regulation, would be catastrophically compressed at hadal pressures. Snailfish achieve buoyancy regulation through a large, fluid-filled gelatinous body cavity instead. Their skulls are incompletely ossified. The bones of the head are not as fully hardened as in surface fish, which researchers believe may reduce the metabolic cost of maintaining skeletal integrity under pressure. Their tissues carry high concentrations of TMAO. But the TMAO relationship creates a biological ceiling. TMAO, at very high concentrations, begins to interfere with osmotic balance, the balance of dissolved substances inside and outside cells that is fundamental to cellular function. Too much TMAO becomes its own biochemical problem. Yancey's modeling of the TMAO data, described in the 2014 Science paper and in subsequent publications, suggests that the maximum viable TMAO concentration in fish tissue corresponds to a depth of approximately 8,400 meters. Above that depth, TMAO is a solution. Below it, TMAO itself becomes the constraint. This would mean that no fish should exist below approximately 8,400 meters, regardless of any other adaptation. The fundamental biochemistry of pressure counteraction reaches its own ceiling before the trench floor does. The deepest confirmed fish observation is 8,336 meters. The theoretical limit is 8,400 meters. The margin between them is 64 meters. Whether those 64 meters represent the actual biological ceiling, or whether some species has found a way around the TMAO constraint and is living in the gap between the current record and the theoretical limit, waiting to be discovered by a lander placed at the right trench at the right depth, is, as of the current scientific literature, genuinely unknown. Jamieson addressed this directly in a review paper published in Deep Sea Research in 2200, and his conclusion, carefully stated, is that the evidence is consistent with a depth limit around 8,400 meters, but that the data are not yet sufficient for certainty. Below 8,400 meters, then, which still leaves roughly 2,500 meters of Hadal Zone above the floor of Challenger Deep, the zone is inhabited primarily by invertebrates, and among them, the dominant animals in nearly every Hadal trench ever sampled are the amphipods. Amphipods are crustaceans, relatives of shrimps and crabs, belonging to the order Amphipoda, and as a group they occupy an extraordinary range of habitats, from freshwater streams to intertidal zones to the open water column to the deepest ocean. In the Hadal Zone, several species have become so specialized for the conditions there that they represent some of the most pressure-adapted multicellular animals on Earth. The most studied Hadal amphipod is Hirondelia gigas, which inhabits the Mariana Trench and several other Pacific trenches and has been the subject of research by Jamieson, by Bartlett at Scripps, and by teams at JAMSTEC, the Japan Agency for Marine Earth Science and Technology, which has conducted Hadal research in the Pacific for decades and whose autonomous underwater vehicles have contributed substantially to the survey of Pacific trench systems. Hirondalia gigas is not large by surface standards, typically between 2 and 4 centimeters in length. It is a scavenger. It feeds on whatever organic material descends from the surface. Arriving at the bottom is what oceanographers call marine snow, the continuous slow rain of organic particles, dead cells, fecal pellets, and biological debris that drifts downward from the sunlit surface layers through the water column. Marine snow is the energy source for essentially the entire Hadal ecosystem. The trench is, in metabolic terms, almost entirely dependent on the surface world above it. Because no photosynthesis occurs in the Hadal Zone, there is no light. And with the exception of any chemosynthetic systems that might exist in some trench environments, a question I will return to. All the energy that powers Hadal life originates as sunlight captured by phytoplankton at the surface and converted into organic matter that eventually sinks. Here is something I did not expect when I began reading about this, and something that seems counterintuitive enough that it is worth stating carefully. The Hadal trenches are paradoxically among the most food-rich environments in the deep ocean. Not because they receive more marine snow per unit of surface area than the surrounding abyssal plain. They receive roughly the same flux of organic particles. But because of their geometry, a trench is a V-shaped depression in the seafloor, and particles settling from the water column above are funneled by the walls of that V toward the axis, the deepest part. Material that would otherwise settle evenly across a flat abyssal plain is concentrated at the bottom of the trench. This funneling effect means that the sediments at the axis of a Hadal trench can have organic carbon concentrations significantly higher than the surrounding abyssal sediments, which supports higher microbial activity, which supports higher populations of the invertebrates, amphipods, polychaetes, holothurians, that feed on the microbes and on each other. The bottom of the deepest place on Earth is, in biological terms, a relatively productive environment. This surprised me. When I looked for it in the literature, I found that it had surprised several researchers as well, including Ronnie Glud at the University of Southern Denmark, whose measurements of sediment oxygen consumption in Hadal trenches, oxygen consumption being a proxy for microbial activity, and therefore for the rate of biological processing of organic carbon, showed rates considerably higher than in adjacent abyssal sediments. Glud's work, published in Nature in 2012 and followed up in subsequent papers, established quantitatively that Hadal sediment microbiomes are genuinely active, not dormant, and that the carbon cycling happening at the bottom of Hadal trenches represents a meaningful contribution to the global deep-sea carbon budget. The trenches are not inert. They are biologically busy by their own standards, in the dark. The microbes in Hadal sediment are collectively described in the literature as piezophiles, from the Greek for pressure, or, more precisely, piezo-tolerant organisms. Organisms that not only survive at high pressure, but function optimally at pressures that would destroy surface organisms. The microbial diversity in Hadal sediments has been explored using genetic sequencing techniques that do not require the organisms to survive collection and return to the surface. You extract the DNA from the sediment, sequence it, and identify what was there by comparing the sequences to known databases. These studies, conducted by teams including Bartlett's group at Scripps and researchers at Jamstec, have found that Hadal sediment microbial communities are distinct from those at shallower depths, not merely a pressure-stressed version of abyssal communities, but a genuinely different assemblage, including archaeal lineages found only at Hadal depths and whose metabolic capabilities are only partially understood. The deep sediment microbiome is, in the language of microbial ecologists, largely uncharacterized. What exactly those archaea are doing at 10,000 meters, what they are consuming, what they are producing, how they are cycling elements through the sediment, is a set of questions that current technology is only beginning to be able to ask. Beyond the microbes and the amphipods, the larger invertebrate community of the Hadal Zone includes sea cucumbers, holothurians, which appear in lander footage at multiple hadal depths and can be remarkably abundant in some trenches, moving across the sediment surface and ingesting it to extract organic material. They have been found at depths exceeding 9,000 meters in the Mariana Trench. Polychaete worms, segmented worms whose surface relatives include the lugworm and the ragworm, have been collected from hadal trenches at depths greater than 10,000 meters. Foraminifera, single-celled organisms that build intricate shells from calcium carbonate or aggregated sediment particles, have been found alive at full hadal depth. And then there are the xenophyophores. I want to spend a moment with the xenophyophores because I think they are among the least discussed, genuinely remarkable things in the deep ocean. And describing them accurately requires dismantling some assumptions so fundamental most people do not realize they hold them. A xenophyophore is a single cell, not a colony of cells, not an organism built of multiple cells in the way that you or I are built of approximately 37 trillion cells. A single cell with a single continuous cytoplasm. That is the standard definition of a unicellular organism. What is not standard is the size. Syringamina fragilissima, the largest known xenophyophore species, can reach up to 20 centimeters in diameter, roughly the diameter of a dinner plate. A single cell. It builds a housing structure called a test, assembled from sediment particles and organic material cemented together by a secreted substance. And it lives on the seafloor, filtering organic matter from the sediment and water. And it is the largest known individual, single-celled organism on Earth. The Schmidt Ocean Institute has documented xenophyophores at multiple hadal depths during recent expeditions, including during surveys of Pacific Trench systems. During the 5 Deeps expedition, Jamieson's team documented them across multiple hadal sites. They are not well understood. Their classification within the eukaryotic tree of life has been revised multiple times. The reproductive biology is largely unknown. How a cell of that size divides, or whether it divides in a conventional sense at all, is among the many questions hadal biology has not yet answered about them. I want to come back to a different kind of light before we continue. Not sunlight, which does not reach the hadal zone. Biological light. The MBARI, the Monterey Bay Aquarium Research Institute in California, whose deep-sea video archive is one of the most extensive in the world, and whose research on bioluminescence has been led for years by biologist Edith Witter, has documented that more than 3/4 of deep-sea animals in the water column produce some form of bioluminescence. In the mesopelagic and bathypelagic zones, bioluminescent light is the dominant mode of communication, predation, and defense. Whether it extends meaningfully into the hadal zone is something the science has barely begun to ask, primarily because most hadal observations have been made by landers operating with artificial lights that wash out any ambient biological luminescence in the immediate vicinity. Some hadal crustaceans are known from related species to produce bioluminescent secretions. Whether the specific organisms of the deepest trenches produce light in their environment is something that, as of the current literature, has not been definitively established either way. It is one of the many questions the zone has not yet chosen to answer. Notice, for a moment, where you are, the ceiling above you, the particular quality of the dark in this room, which is not the absolute dark of 10,000 meters, but is, for this hour, a reasonable stand-in. Your breath, which is moving in and out at a pace your body has set, without you asking it to. Your body, which is heavier now than it was when you lay down, the day releasing its hold in the way it does in the hours before sleep. A significant fraction of the oxygen in that breath was produced by the ocean, by phytoplankton in the surface layers of the sea, photosynthetic organisms so small that a single drop of seawater at the right latitude and season can contain thousands of them, converting sunlight into organic matter and releasing oxygen as a byproduct. Oxygen that moves into the atmosphere and eventually into your lungs. Not as a metaphor, chemically, biologically. The oxygen in the room where you are lying came, in substantial part, from the sea. The ocean covers 71% of the planet you are lying on, and it is producing the air you are breathing right now. And it has depths you and I will never visit. And most of what lives in those depths we have not yet seen. Your only job right now is to be horizontal. Everything else is optional. Now I want to spend some time in the territory that I find most interesting, which is the territory of what we genuinely do not know. Because the history I have been describing, the Trieste, Cameron, Vescovo, the snailfish, the amphipods, the xenophyophores, represents the known, what has been observed, sampled, measured, published. And the known in Hadal biology represents a small fraction of what is very likely there. Consider the basic coverage question. Of the approximately 37 Hadal trenches on Earth, how many have been visited by any instrument at all? A camera, a lander, a submersible, at their deepest points? Jamieson and colleagues, reviewing the state of Hadal exploration in papers published across the past decade, have noted that a significant number of Hadal trenches have never had an instrument deployed at their axis. Several more have had deployments only at their shallower margins. The total number of trenches with any sampling at confirmed full depth is a minority of the 37. Of the volume of water and sediment that constitutes the Hadal Zone globally, the proportion that has been directly sampled is, as NOAA has repeatedly stated in its public exploration documentation, genuinely small. We know that the Hadal Zone contains life. We have a reasonable picture of the major taxonomic groups present. We have essentially no idea of the biodiversity at the species level, how different the communities are between trenches, or what ecological processes operate in the zones of each trench that have not been visited. The Kermadec-Tonga Trench System in the Southwest Pacific, which extends to approximately 10,800 meters at its deepest point and runs for roughly 2,600 kilometers, one of the longest trench systems on Earth, provides an instructive case. It has been studied more thoroughly than most Hadal trenches, with multiple lander deployments and some crewed submersible time. The biology found there, particularly the amphipod community, is substantially different from the Mariana Trench community, despite similar depths and similar pressure conditions. Different species compositions, different relative abundances, different community structure. The Kermadec-Tonga system is at a different latitude, with different oceanographic productivity in the surface water above it. It has a different geological history. The specific reasons for the differences in community composition are not well understood. Whether those differences reflect general ecological principles that would allow prediction of community structure in unvisited trenches, or whether each trench is essentially its own biological island, assembled by its own particular history of geography, oceanography, and chance, is an open question that Jamieson addresses directly in his book on the Hadal Zone, published by Cambridge University Press in 2015, without reaching a settled conclusion, because the data are not yet sufficient to settle it. The chemosynthesis question is one I kept returning to during my research, and it remains, I think, one of the more interesting unresolved issues in Hadal science. At hydrothermal vents, seafloor hot spring systems, first discovered at the Galapagos Rift in 1977 by a team including geologists Jack Corliss and Robert Ballard, diving in Alvin. There are organisms that derive their energy not from organic carbon produced by photosynthesis, but from the oxidation of inorganic chemicals, primarily hydrogen sulfide. Chemosynthetic ecosystems of this kind are independent of the sun. They require no marine snow. They are metabolically self-contained, running on the chemistry of the Earth's interior. Hydrothermal vents have been found at mid-ocean ridges at depths of roughly 2,000 to 4,000 meters. Have they been found in Hadal trenches? The answer, as of current literature, is possibly in limited forms. There is geological reason to think that some hydrothermal activity might occur in certain Hadal trench environments because trenches are formed at subduction zones, active plate boundaries with significant geological energy. Some fluid seepage has been documented in trench systems. Cold seeps, areas where fluids enriched in methane and hydrogen sulfide move slowly from sediment, have been documented in some Hadal environments, including in the Japan Trench, and do support small chemosynthetic communities. But the dense, biologically rich hydrothermal vent communities of tube worms and vent crabs and vent shrimps, characteristic of mid-ocean ridge systems, have not been confirmed in Hadal trenches. The geological conditions differ. At a mid-ocean ridge, new crust forms and magmatic heat drives vigorous hydrothermal circulation. At a subduction trench, crust descends, and while there is some thermal and fluid activity, the conditions for the concentrated stable venting that supports full venti ecosystems may not be present in the same way. Whether this absence is fundamental or simply reflects the fact that most Hadal trenches have never been searched, for venting is one of the many questions that requires more dives to answer. The microplastic question brings the deep ocean and the surface world into the most uncomfortable proximity. And it is worth describing in detail because the science is recent and specific, and does not allow easy interpretation. Alan Jamieson and colleagues, in a paper published in Royal Society Open Science in 2019, reported on the presence of microplastic particles, fragments of synthetic polymers, fibers from textiles, in the gut tissue of Hadal amphipods, collected from 6 different trenches: the Mariana, the Tonga, the Kermadec, and 3 others spanning the Pacific. The amphipods were collected at depths ranging from 7,000 to over 10,000 meters. Every single animal tested contained microplastic, 100%. The most abundant polymer identified was PET, the material used in plastic water bottles and synthetic clothing fibers. The concentrations found were higher than those measured in some near-shore marine environments. Jamieson and his co-authors are careful in their language. The ecological and physiological effects of microplastic ingestion at these concentrations in Hadal animals are not yet understood. This is not a paper arguing that the Hadal Zone is dying. It is a paper reporting what was found in the stomachs of animals at the bottom of the ocean. And what was found is a record of the surface world. The trench collects everything that the water column above it delivers, and the water column above it collects everything that enters the ocean. The geometry that concentrates marine snow also concentrates synthetic particles. Every piece of plastic that enters the ocean eventually, through a chain of circulation and sedimentation that operates on timescales of years to decades, arrives somewhere on the seafloor. Some of it arrives at the bottom of trenches. The question this raises is one I cannot resolve, and am not going to pretend to resolve. It is the question of what it means to call a place remote when no barrier, physical or chemical, actually separates it from anywhere else. I have been sitting with that question throughout this research. I still do not have an answer that satisfies me. I am not sure one is available. There is one more thread I want to follow before we begin the slow return to the surface, And it is the thread that takes us, counterintuitively, and by a chain of scientific reasoning that I think is genuinely compelling, off the planet. In 1977, the team aboard Alvin, who descended to the Galapagos Rift, found not just hydrothermal venting, but a community, a dense, visually striking community of life clustered around the vents. Giant white clams, colonies of mussels, pale crabs, and rising from the vent structures in clusters, tubes of white and red up to 2 meters long. The vent tube worms, Riftia pachyptala, had no mouth and no conventional digestive tract. And when researchers studied them in the years after the discovery, they found that the worms derive all their energy from symbiotic bacteria living within a specialized internal organ. Bacteria that oxidize hydrogen sulfide from the vent water and produce organic carbon by chemosynthesis, feeding the worm entirely from inside. No sunlight, no photosynthesis, no marine snow from the surface. A complete ecosystem running on the chemistry of the Earth's interior in total darkness at 2,500 meters below the surface of the Pacific. John Corliss, one of the scientists aboard Alvin during those initial dives, described the moment in subsequent interviews as one that changed what he understood the word life to mean, not in a poetic sense, in a definitional sense. The discovery demonstrated that photosynthesis is not the only pathway to a complex multicellular ecosystem, that you do not need the sun, that the energy of the Earth's interior, expressed as heat and chemical gradients, is sufficient to power biology. The implications for NASA's astrobiology program, the field concerned with understanding the distribution of life in the universe, were immediate. Europa, one of Jupiter's 4 large moons, was already suspected on the basis of spacecraft flyby data to contain a liquid water ocean beneath its ice shell, one heated by tidal flexing from Jupiter's gravity. Enceladus, Saturn's small ice-covered moon, was confirmed by the Cassini spacecraft in 2015 to have active hydrothermal venting on its seafloor and to be ejecting water vapor and organic compounds into space through cracks in its surface. Both moons have liquid water, internal heat, and chemistry. None of that chemistry is driven by sunlight. All of it is analogous, in principle, if not necessarily in detail, to the hydrothermal systems of Earth's mid-ocean ridges. NASA formalized this connection through an initiative called SUSA, Systematic Underwater Biogeochemical Science and Exploration Analog, which uses deep-sea hydrothermal environments on Earth as scientific analogs for the subsurface ocean environments of Europa and Enceladus. The researchers involved described their deep-sea work explicitly in those terms. They are studying Earth's deep ocean in order to develop the science and the methodology that will be needed, eventually, to look for life on ocean worlds. The Hadal Zone itself is not the primary analog. The vent systems are closer to what the ocean world interiors might resemble, but the Hadal Zone contributes something the vent systems alone cannot provide. Evidence of multicellular organisms surviving and functioning at pressures more extreme than anything currently modeled for Europa or Enceladus. The Mariana snailfish, hunting and reproducing at 8,300 meters under 833 atmospheres of pressure, is data. It is a data point about the upper range of pressure tolerance in vertebrate life. It is relevant to any model of what life might do in an ocean on another world. I want to say something about the whale falls before we begin to surface, because they sit at the intersection of several of the threads I have been following tonight. The marine snow system, the chemosynthesis story, the question of what counts as isolated and what counts as connected. A whale fall is what happens when a large cetacean dies at sea and sinks to the deep ocean floor. Blue whales, fin whales, and sperm whales can reach between 20 and 30 meters in length and masses up to 150 tons. When they die, they sink. The descent through the water column takes several days. At depth, the body arrives on the seafloor as one of the largest single pulses of organic carbon a given area of deep seafloor is likely to receive in any given decade, equivalent in organic energy to approximately 2,000 years of background marine snow falling on a comparable area. The successional community that colonizes a whale fall was first characterized in the 1980s by Craig Smith at the University of Hawaii, who has studied whale fall ecology extensively and whose work established the successional stages of whale fall communities in the Pacific. The first stage, lasting months, is dominated by mobile scavengers, sleeper sharks, hagfish, rat-tail fish, and crustaceans that strip soft tissue from the carcass. The second stage, lasting years, is the enrichment opportunist stage, in which polychaete worms and crustaceans colonize the exposed bones and surrounding sediment, feeding on remaining organic material. The third stage, lasting decades, is the sulfophilic stage. Anaerobic bacteria in the bone begin breaking down lipids in the bone marrow, producing hydrogen sulfide as a byproduct, which supports chemosynthetic organisms, microbial mats, small tube worms, and clams related to the species found at hydrothermal vents that derive their energy from sulfide oxidation. A whale fall becomes, in its final stage, a small temporary chemosynthetic island on the deep sea floor, supporting organisms that would otherwise require a hydrothermal vent to survive. Whether whale falls occur in the Hadal Zone, whether the geometry of whale mortality in the open ocean and the locations of hadal trenches occasionally result in a carcass sinking into the trench itself, is probable but has not been directly documented. What the successional community of a hadal whale fall looks like, whether the extreme pressure modifies the biochemistry of decomposition or the species composition of the scavenger community, whether the chemosynthetic stage proceeds similarly to an abyssal whale fall. These questions have not been answered because a hadal whale fall has not been studied. One more item on the list. There is a question I keep returning to, and I want to ask it to you directly. We have been in space for 65 years. We have sent probes to every planet in the solar system, landed on the moon 12 times with humans, pointed the Hubble and Webb telescopes at galaxies 13 billion light-years away, and are seriously planning crewed missions to Mars. In that same 65 years, the deepest point of our own ocean has been visited by crewed vehicles fewer than 30 times. Mars has received more than 50 robotic missions. Challenger Deep has received 3 crewed submersibles. The Moon is 384,000 kilometers away. Challenger Deep is under the Pacific. Why does the direction of our curiosity point so persistently upward rather than downward? I am not arguing that space exploration is the wrong choice. I have spent nights on this channel following questions about stellar death and black hole physics and the formation of galaxies, and I will spend more nights doing so. The question I am asking is different. It is a question about what we find legible as frontier. Space has a horizon you can look at. It has an image, a small blue marble against black, that reorganizes the way you see yourself. The deep ocean does not offer that image. It offers sediment and pressure and something pale moving in the dark at the edge of your lights. Whether that is a less important frontier or simply a less photogenic one is something I genuinely do not know. I would love to know which way this lands for you. Leave a comment if you are still awake, or come back to it in the morning. It is the kind of question that does not get smaller if you leave it alone. We are coming back to the surface now, slowly. The way the Trieste came back, patient through the pressure gradient, through the darkness that became slightly less dark, through the indigo of the mesopelagic, through the blue that began to have some relationship to the sky above it. The water in your body, approximately 60% of your total mass, has been through the ocean. This is not a metaphor, and it is not a spiritual claim. The water cycle that fills the glass on your nightstand, that runs through your circulatory system, that maintains the osmotic balance of every cell in the brain, following these words, is the same cycle that draws water from the ocean surface into the atmosphere as water vapor and returns it through rivers and rain. The hydrogen atoms in the water in your cells have cycled through evaporation and condensation and precipitation and groundwater and river flow and ocean over and over for billions of years. Some of the water in your body was, at some point in its history, in the deep ocean. The boundary between you and the sea is a boundary of form and of time scale. It is not a boundary of substance. And at the bottom of the ocean right now, as you lie in the dark and your breathing slows to the pace that sleep uses, there are snailfish moving across pale sediment at 8,000 meters. Their tissue full of TMAO, their incompletely ossified skulls holding their shape under 800 atmospheres. There are amphipods clustering around organic matter that descended from a world they have no concept of. There are xenophytophores, 20 centimeters across, a single cell doing what a single cell of that size does, in the cold and the pressure and the complete dark. There are microbes in the sediment cycling carbon that began as sunlight weeks or months ago at the surface of the sea. There are things we have not yet seen and have not yet named. The Hadal Zone refuses to give up its secrets, not because it is hiding them. It refuses because we have barely asked. There is still most of it left, most of the trenches, most of the species, most of the bottom. The zone has been there for tens of millions of years and will be there for tens of billions more. Completely indifferent to whether we visit it or not, running its own chemistry in the dark, patient in the way that only a place that does not experience time the way we do can be patient. You do not need to carry any of this to morning. Nothing was tested tonight. Nothing needed to stick. If what happened in these hours was only that you lay in the dark while a voice described a world you will never visit, and your body found its way toward rest, that is enough. That is more than enough. The Trieste came back. Cameron came back. Walsh came back and lived another 63 years and gave interviews well into his 80s about what he had seen on the floor of the Mariana Trench in 1960. Vescovo came back. Dove, again, came back again. They all carried small pieces of the zone back to the surface with them. Sediment samples, measurements, footage, and a quality of knowledge that is different from the knowledge you get from a paper. Different even from the knowledge you get from footage. The knowledge of having been there, of having descended 5 hours into the dark and arrived somewhere that, when you turned on the lights, turned out to be occupied. The snailfish were there before we knew they existed. They are still there, moving in the dark at 8,000 meters, indifferent to our arrival and to our leaving, unchanged, as far as we can tell, by the few brief moments when a cone of light from a human machine illuminated the seafloor beside them. The zone holds most of its secrets still. It will be there tomorrow night, and the night after that, and for as many nights as it takes. It is not going anywhere. The ocean is very old and very full, and in no particular hurry. And somewhere far below the surface of the world you are sleeping in the cold and the dark and the quiet, something is alive that does not know your name. And that right now is exactly as it should be.